Good afternoon to Dr. Ruben and to Dr. Ong. My name is Vijay Sivanaran, and in this presentation, I'll be basically giving you all a critical review of my research paper. The title of the research paper is Successful Development of Bacteriocins in Therapeutic Formulation for Treatment of MRSA Skin Infection in a Murine Model. So, firstly, I'll be talking about what MRSA is. Methicillin-resistant Staphylococcus aureus is one of the most common and virulent strains of antibiotic-resistant Staphylococcus aureus. MRSA has been identified as a high priority disease on the World Health Organization's global priority pathogen list of antibiotic resistant bacteria for antimicrobial research and development. So now I'll be talking about the causative factors of MRSA. The primary causative factor of MRSA is antibiotic resistance. MRSA is the result of often unnecessary antibiotic use. For years, antibiotics have been prescribed for colds, flu, or other viral infections that don't respond to these drugs. Even when bacteria are used appropriately, they contribute to the rise of drug-resistant bacteria because they don't destroy every germ they target. Bacteria live on the evolutionary fast track, so germs that survive treatment with one antibiotic soon learn to resist others. So this is the primary causative factor of antibiotic resistance of MRSA. The second causative factor of MRSA is the transmission of MRSA. Exposure to contaminant high-touch surfaces or fomites and close contact with colonized people or animals in this environment may contribute to CA-MRSA transmission, which is CA refers to basically community-acquired MRSA transmission. As a result, this will then lead to more individuals having the disease. Following that, now I'll be basically talking about the physiological disruption for MRSA. In the community, MRSA most often causes skin infections. In some cases, it may cause pneumonia, which is lung infection. The other clinical signs and symptoms that can be caused by MRSA are the following, which are chest pain, cough or shortness of breath, fatigue, fever and chills, feeling unwell and headache. So these are basically the physiological disruptions for MRSA. Now, I'll be talking about the novel treatments for MRSA. There are three normal treatments for MRSA. The first treatment is a ciprofloxacin modification with a triphenylphosphinium moiety that can be used to target bacterial membranes through ester and amide coupling. So the second treatment for MRSA is by the usage of Belcecon C antibiotic that helps by targeting the bacterial cell membranes. And the third novel treatment for MRSA is by using bacteriocins. So these are the three novel treatments for MRSA that can be used to treat this particular disease. Moving on, now I'll be talking about the major findings of the research paper. The first major finding of the research paper is that in the results, strong synergy was found in the combination of PenG, GARX, and MP1 with lowered MIC of PenG, MP1, and Garvacin KS from 2,500 microgram per ml to 8 microgram per ml, from 2.5 microgram per ml to 0.08 microgram per ml, and from 32 microgram per ml to 4 microgram per ml, respectively, by using checkerboard assay and AAMP. The second major finding of this research paper is that with the help of HPC, MIC of GARK-KS and PNG and MP1 was further lowered into 2 microgram per ml and 40 microgram per ml, respectively. The third major finding of this research paper is that fusidin cream as a commercial drug used for fusidin cream is one of the commercial drugs that is used for skin MRSA infection, and it's also a positive control, and it has been compared with the formulation in agar diffusion method. And results have showed that there's a development of resistant colonies in fusidin inhibitory zone, but it is not found in the formulation. The fourth major finding of the research paper is that the results from the murine skin model experiment have showed high reduction of bioluminescent signals in murine treated with formulation in day 2, followed by the reappearance of the signals in day 4 under single treatment. By changing the treatment schedule, it is proven that the long-lasting effect from formulation is produced in the schedule of 4 days treatment. So these are the major findings of the research paper. Now, following this, I'll be talking about the major weakness of the research paper and the suggestion to improve it. The primary major weakness of the research paper is that the researchers failed to assess the toxicity effects of the formulation. Hence, psychological effects are unknown in regards to the formulation treatments against MRSA. The suggestion that can be done to improve this mistake is pharmacokinetics profile, immunogenic response, human hemolysis assay, biochemical and histopathological analysis can be used to assess the toxicity effects of the formulation. It is imperative to evaluate toxicity effects of the formulation, as it may lead to potential effects such as genotoxicity, carcinogenicity, immunotoxicity, and organ impairments in living organisms. So this is the first major weakness of the research paper. The second major weakness of the research paper that is in the study that it was mentioned that the formulation was not fully optimized. As the release of drug component was fast, this will then cause dilution before all the bacteria was killed, thus presenting a shorter lasting effect. The suggestion that can be done, the suggestion that I can imply to improve this is to basically, the researchers may consider using same medium for fusidine. So this is the second major weakness of the research paper. And the third major weakness of the research paper is that box plot was solely used as a means of statistical analysis in the study. Hence, deficit inference was drawn from the data. The suggestion to improve this is the suggestion to solve this problem is that a t-test could be conducted to compare the formulation group with the untreated group and fusidic acid group, so that the presence of significant difference can be determined. So these are the few major weaknesses and the suggestion that can be done to improve this research paper. Here are the references that I use for this review. Thank you very much and have a great day.